what is cell cycle regulation and how it is affected by different proteins or enzymes. Basically, when we talk about cell cycle, we say that it is highly regulated. Highly regulated means that it will perform in normal functioning based on specific phase order. For example, most important phase about cell cycle is S phase, which is synthetic phase. So before S phase, we have other phases such as G1 or G2. G1 is basically pre-synthetic phase and the G2 is post-synthetic phase. In between G1 and G2, we have S synthetic phase. So what happens in these phases is that we say that S phase is the most important. Why? Because it is the phase while there is synthesis of DNA and then replication of DNA is also occurring during this phase. So for that, this synthesis and replication, there is need for some helping materials, and those helping materials are basically two, which are cyclins, which are proteins which are used for progression of cell cycle towards this regulation, and this progression is done by cyclins with the help of CDKs, which are basically cyclin dependent kinases. These CDKs are basically enzymes which help protein cyclins, and these cyclins plus CDKs, they help the cell in regulation mechanism. So what happens is these cyclins and CDKs, they help the cell cycle to make decisions at specific time and point. So what is the importance of this time and point? We will look into the next slide, which is that cell has to make two decisions, either to divide or not to divide. So this decision of division or not division, it should be at some specific time and at specific location. For that specific location, basically that is called a decision point, and this decision point in cell cycle alone is called restriction point. So if what happens if this cell is damaged or if there is abnormality, then what will happen at this restriction point, the cell division will stop, so that there is no cancer being generated. Because if a normal cell division occurs, then it will propagate in the next cells. So for that purpose, if there is some abnormality, then this cell cycle is arrested. Arrested means stopped for recovery. So here I have given examples of some cyclins and their CDKs. For example, if I talk about different cyclins and different things, first of all I will start from this R. This is basically our decision point or restriction point, which helps us to identify if the cell division will occur or no. Or similarly, if we have different parts such as these CDKs, so if I start from this CDK2, which is a partner of cyclin B. So CDK2 is our enzyme and cyclin B is our protein. So they are in combination to each other. And this CDK2 and cyclin B, they are part of this mitosis phase, M phase. If we move toward this G1 phase, we have CDK4, which can be partner of cyclin D1, 2 and 3. Similarly, then we have CDK2, which has the partner cyclin E, and so on. So what happens? We have these DNA damage points. So what happens if there is some damage in DNA, then they will stop further partitioning of the DNA here, so that the cell remains in the stable stage. So what happens, or what is particular interest to us in this particular slide, if there is some tumor inside the cell, what will happen? So what will happen will this R, basically this restriction point, it will becomes abnormal or unoperational. So if this is abnormal or unoperational, what will happen? That it will not be able to identify decision point, and then what will happen? This cell growth will become abnormal. कि ये जो R है अगर अनऑपरेशनल हो जाए या ये अगर अपनी फंक्शनिंग छोड़ दे, तो जो सेल ग्रोथ है वो नॉर्मल नहीं रहेगी, अनकंट्रोलेबल हो जाएगी, जिसकी वजह से इसमें खराबियां पैदा होंगी. So what we say, if cell is damaged by chemical or some other chemotherapeutic events, what will happen? Cycle is arrested prematurely in order to allow time for recovery from damage. So here I have given an exact example of cell cycle regulator. For example, as I said, that they are basically different proteins which are called cyclins. So what happens in this regulatory proteins? So here we have given an example of cyclin B1, which is basically as shown in the previous diagram also. It is expressed at time of entry of cells from GT to M phase. Express से मुराद क्या है कि एक्टिव होता है उस टाइम, and when does it destruct itself? At the time of exit from M phase. So इसकी entry के वक्त ये generate होता है और exit from M phase it destroys or destructs. So here we have given an example of activation and destruction. So what is happening here? So whenever we taking these thick lines, which are basically these thick lines, so what is happening in these thick lines? They show those things which are enhanced activity of CDKs. कि जहाँ पर CDKs की activity enhance हो रही है वो thick line से show हो रहा है, और which are these thin lines, they are shown by basically inhibition of activity, यानी कि उनमें activity जो है वो reduce हो रही है. So what does this complete figure show us? This figure show us activities of various CDKs and their cyclins. So as shown, we can see that about this CDK4 cyclin D or CDK6 cyclin D, they are active during the middle of this G phase, and then they are very active until here, and then they start to die out, and they die out during this R phase, end of this R phase basically. So this is shown by this thing. Then we have this, suppose if I take example of CDK2 cyclin A, it start of this S phase, it gets on concentrated, it reaches this point, and then it starts to dry out and it reaches its end towards the end of this S phase.